thought I will begin my presentation with a small story, which is when we were PhD students. And you see this beautiful building where you are, this is given to us by Department of Biotechnology. And when we were PhD students, there were no Department of Biotechnology. So we really worked in resource poor settings. To give you an example, our fellowships were like 400 rupees a month. At AIIMS, both MD and PhD students, they used to get 400 rupees a month. The government of India decided to increase the fellowship to 750. So we were all very happy. But then AIIMS decided to only increase the salary of MDs and not of PhDs. So we, of course, we were on strike for like 67 days and no work in the lab. And you could imagine my PhD thesis was on drug addiction, and we had neuronal cell lines, which would have receptors for morphine to study drug addiction. We lost all our cell lines. There was nothing left. Anybody heard of Marshall Nirenberg? No? Hargobind Khorana? Yes. So Marshall Nirenberg and Hargobind Khorana, they shared the Nobel Prize for deciphering genetic code. Marshall Nirenberg had these cell lines, but to call him, during those days there were no cell phones, 3-minute call to US would cost you 100 rupees, which is 25% of your monthly salary. And you go and stand in Eastern Court somewhere in Connaught Place, and when your number comes, Indians talk very fast. So you go through like, I am so and so and I need this and this and this and this, because you are running against the time, and from the other side you hear hello and your 3 minutes are over. So now you don't have, and there is no support from the lab. This money is coming from your pocket. To an extent that now if I can get my experiment done in US, and if I see the results are not as I expect, I can fly from here within 24 hours, reach, that's the kind of difference has come. So I really thank Department of Biotechnology for changing the face of biotechnology in this country. They have been the oxygen here. But looking at AIIMS, we learned so much in those resource poor settings. If you can do something there, you can do anything anywhere. So next slide would show some of my PhD students. I have had 29 PhD students. I have been working at ICGEB for 27 years, and without them, means I am useless. They are the spinal cord, they are the brain of the lab, and they are so well committed. They have a lens in their hand of so what next. Each one of them has been taught in our lab that if your work has an impact, you don't need to factor it. Most of us are looking at impact factors of our publication. None of them are looking at those things. They want to make a difference. So I actually salute each one of them for coming to our lab with self-determination. And we have 23 diagnostic kits in the market, and these are not boxes. They are the world leaders when they really sells in millions. And you can't assemble these kits by hand, you need robots to assemble them. So I am showing you about 6 or 7 or 8 of these diagnostic kits here, and Government of India recognizes these efforts. So it's not that what you do for public health, if you don't publish, nobody recognizes. That is not true. I thought for today's presentation, and I am trying to condense story of about 25 years in probably 10, 20, 15 minutes. So I would give you some flavor of what we do in terms of diagnostic highlights. I thought we have diagnostic tests for hepatitis B virus, hepatitis C virus, HIV, dengue, to name a few. And I am sure all of you, whether you are biologists or not, you know these names. We have a dengue vaccine candidate, which I will give you a small flavor of it. And anyone have suffered from dengue before? Yes? So I have had dengue, chikungunya, malaria. So whole MCD is inside. MCD full form is malaria, chikungunya and dengue. So we have botanical drugs against dengue. and then I will show you how is this possible to collaborate with various institutes and various companies to make these things happen. Now, this is I am storytelling you about 15 or 17 years back. You know, hepatitis B virus, according to WHO, kills more people in a single day than HIV kills in 1 year. But do we hear about hepatitis B virus? No, we hear about HIV all the time. HBV is a silent killer. You know, hepatitis B virus infects your liver. Liver is the biochemist, and biochemists are the best people. So, liver does all the job. One liver can take care of five persons. So, which is a good thing, but the bad thing is that unless it is 80% damage, you won't know, there won't be any clinical symptoms. So, hepatitis B virus infects liver and cause liver cirrhosis, and there was no test to detect it. The tests which were available were very expensive, and I am again talking about 17 years back, where China was trying to get into our country, and many of the Indian diagnostic companies would always assemble things. You know, we never make anything, we assemble, we buy things from outside, assemble them together and then put them in the market, and we say that this is ours. So, when some of these companies had gone to China and they had seen, the Chinese said that we will fill Indian market, diagnostic market with so much of our products, you won't know what happened. Now their survival is at problem. Now they come to various institutes, and one of that happened to by ICGEB, and we gave them an antibody produced by a PhD student in our lab. And this antibody recognizes hepatitis B virus, but it was so good that instead of, you know, whatever amount they were using like a microgram, for this antibody you could make a test with 2 nanogram, which meant from the same amount of biological material you could make 500 tests. Production cost of this is less than this bottle water. 6 rupees is the production cost, and this sells globally. WHO is the major buyer of this test. I thought I will show you a small video that how simple are these tests. You know, they are point of care tests, they are not very complicated. This is just a one minute video. What happens in this is that everybody have seen a dipstick, right? Dipstick is a small stick which has all the, just like a pregnancy test. It's a small stick, and on this side you just you basically open the pouch, add a drop of serum or plasma. Nowadays you can even add whole blood from the finger prick, and then just wait for 15 minutes and you will start to see red lines appearing, which was in the videos were much more clearer. What I am trying to show is an antibody here against a hepatitis B virus. This antibody produced in ICGEB, the clone is given to them, and this antibody will recognize the virus, and these antibodies are gold labeled. So, they have a red color on them, and when they move towards this side, this is the same antibody which is unlabeled. So, what you get is a sandwich between the two antibodies. The hepatitis B virus is trapped and it gives you a red color here, and since all monoclonal antibodies are made in mice, these are anti-mouse antibodies as a control line. So, at the end of this test, which involves just adding 2 drops of plasma or serum or blood, walk away, do nothing for 15 minutes, come back and look at the test. If it gives you 2 lines, you have hepatitis B virus infection. If it gives you 1 line, you do not have a hepatitis B virus infection. It gives you 0 line, it means the test has gone bad. So, all the controls are built in. It is a simple assembly line. These lateral flow assays have been in the market for 40 years, and you may have seen in IIT at many places we make MEMS, we make microfluidics. These old technologies die hard, it is very difficult to beat them. So, from hepatitis B, when we had such a grand success, now these companies were addicted to ICGEB, they thought that whatever we give it to them, it is worth something, it is not trash. That was another, we were working with All India Institute of Medical Sciences, another hepatitis, hepatitis is called by hepatitis A, B, C, D, E, F, G, number of viruses are growing up. Both of them are transmitted through blood. So, hepatitis C virus, we will work with clinician at AIIMS, and they will tell us that, Navin, we see a patient who clinically looks like suffering from hepatitis C virus. When we do the test, test is negative. So, something is wrong with the test. And what is wrong with the test? Because hepatitis C is a RNA virus. RNA viruses keeps changing, and if you look at globally, there are 6 genotypes and 92 subtypes, 1A, 1B, 2A, 2B, 3C, 6A, 7, 8, 9, so many of them, and they have distinct geographical distribution. The test during those time would come from US to India. So, the subtype of the virus which is in India is very different than what is in US. So, you would miss certain cases, about 3 to 5 to 7% of those cases, but clinically they are all hepatitis C. And there were kits available in India, and hepatitis C virus has 10 proteins. So, when you have infection, you make antibodies to many of these proteins, and if you have to capture these antibodies, you have to use 5 or 7 of these proteins to capture those antibodies. It means 5 fermentations, 5 purifications, or during those days you do not do any of these things, you take these proteins from 5 different companies. We were working on these things to make them one protein. Can one protein do the job of 5 proteins? And that was the time there was a company, Tulip Lab in Goa. They were making this hepatitis C kit very successfully. One protein come from Australia, one come from Italy, one come from somewhere US and some place Brazil, and one of the company now decides not to give you one component of that test, because the company is bought over by Japan and they say we do not give this protein to India. Now, a working test stopped. They come to us, they say, can you make this protein for us? We said, no, we can't make this protein for you. Why don't we free you from all clutter? Instead of importing 5 proteins, I give you one magical protein which will do the job of 5 proteins. So, we were designing a single multi-epitope protein, and while as I told you, you have to maintain the sensitivity, specificity and worldwide, because there are 92 viruses, you have to detect each one of them. So, here is an example that instead of showing you 7 proteins, I am just showing you 3 proteins. So, you see these arrows which shows you the linear immunodominant epitopes, the hot spots of these proteins. So, what we do, these are HCV specific epitopes, these are linear, and immunodominant means we make antibodies to them. So, we kind of take the hot spots of these proteins, and through our genetic engineering we align them, we put them into expression vector and make a protein which we call multi-epitope protein. This is an assembly line, you could do it for HIV, for dengue, for syphilis, or whatever. you could replace 5 proteins with a single designer protein. This is where the PhD students come in, and they identify these, these epitopes, and then they link them together, and then they make a protein, and then they check the sensitivity, specificity, and if it is not good, then we go back, and these are modular design. You can keep adding more epitopes to them. And so you have pathogen specific. I have kind of made it like HIV, HCV, dengue, syphilis. You can imagine anything. Linear immunodominant epitopes like these. You put them glycine linkers. Glycine has no side chains. So it's like a bracket. So everything is moving separately. And you put them in through a DNA technology into an expression vector. You have a single protein doing the job of 5 proteins. So I think this was a great success. When we gave this protein to that company, in fact, they came back to ICGEB saying that they had 44 samples of HCV infected serum which were not detected by other tests, and they were able to pick up by this. So why we do multi-epitope designer proteins? One, of course, I told you, higher sensitivity. Why is it higher sensitivity? Because the whole protein is useful hotspots. Everything is immunodominant. Higher specificity because if you can get rid of cross-reactive epitopes. Sometimes viruses, for example, flaviviruses, HCV, dengue, they mimic. You have similar epitopes. So you could cross-react with each other. Here, like a tailor, you could remove those cross-reactive epitopes. You could directly capture antibodies like a sandwich. You could put a multi-epitope protein on this side and the other side, and put an antibody in between and get a sandwich. In a shake flask, in a small shake flask in the lab, you make enough protein to make 50,000 tests. You do not have to do multiple peptide synthesis or multiple expressions and purification of proteins, and you could detect more than one infection. You could have a multi-epitope protein for HIV, for HCV, for dengue. You could do many of those. Multiple serotypes, I told you, you could have like 92 subtypes. You could look for the conserved immunodominant epitopes and make a protein out of that. It looks very simple, but anything which looks simple is much more complex. It requires the knowledge of pathogen specific linear immunodominant epitopes. This is where I salute my PhD students who keep on looking at those epitopes. And that's a separate session. How do you do that? And this was the FlaviCheck with whole blood HCV infection. Now, instead of doing with those 5 or 6 proteins, they were just doing it with 1 protein here. So this was a success story. And you can go on. So I am not boring you. I showed you one for hepatitis B virus, which was an antibody. I showed you one for hepatitis C virus, which for which we had made an antigen. Here is a HIV for which you need antigen and antibody detection. So it's a similar assembly line. So now we have a HIV detection, HCV infection, HBV infection. Anybody has donated blood in blood banks? Right? So what do they do? If you look at WHO, it says roughly that about 80 million units of blood is collected globally with about $8 billion cost. And only 80% of the world has access to only 20% of the safe blood supply. More than 50 countries do not have quality system, so the blood is transfused without checking. I have known my friends who have got hepatitis C infection through blood transfusions. So if any one of you have donated the blood, first thing they do is they put you on a chair, take 300 ml blood out of you, and then they do HIV, HCV and HBV test. And then of course you have a lot of medical waste and there is a small amount of safe blood. But what happens, what we are proposing is that I take a finger prick from you and pre-screen instead of post-screening. I do a test in 10 minutes while you are waiting. I know whether you have infection for HIV, HCV or HBV. If yes, I do not collect your blood. Right? So this is a 3-in-1 test which decreases the production cost. You do not have to do too many tests. It streamlines the collection process. You do pre-screening. It reduces the cost related to unsafe blood collection because that is a biological waste. You have to throw it somewhere, and can be used at collection camps in remote areas. It is very simple test. Once you have this good beautiful designer antibodies or antigens which are diagnostic intermediates. Thanks to Professor Bhan and Department of Biotechnology, we collaborated with University of Turku. Turku, you would probably not understand, it is a small city in Finland, but when it comes to diagnostic, it is a Stanford of diagnostics. 180 people in one department work on diagnostics. If you tell them to work on drug or vaccine, they would not touch it, but on diagnostic, that is the focus. In our whole country, we do not have 180 people who work on diagnostics. So, that is the lens, that is the focus, and thanks to Professor Bhan, because they do not publish their work in high impact factor 0.6, 0.4, we are just married to high impact factor all the time. So, we would have never read their paper, but once you go there, their PhD students are just soaked into this kind of work. After their PhDs, they do their postdoc in companies, they do not do postdoc in the same universities, and many of them open their own companies. So, you could have advanced diagnostic concepts wherein you could dry all your reagents in a well. You start with the addition of a blood sample, mix and read, mix, wash and read. So, there is a wash step, it gives you a 2 nanogram per liter sensitivity. You can use nanoparticles, it can give you 0.04 nanogram per liter, but the best is this, that you could use upconverting phosphors. These are new fluorescent molecules which can be stimulated by infrared, and blood does not absorb infrared, rest everything is UV. So, there is no autofluorescence, so there is no wash step. You can use whole blood, basically you add your reagent to the blood, mix and read, there is no wash step. With whole blood you can do, and the sensitivity is 1 femtomole per test, which is almost reaching PCR sensitivity without doing PCR. So, it is the ease of immunoassays, sensitivity of PCRs. So, I think we have made sufficient amount of diagnostic proteins, and all of these proteins and the clones, they are in Finland. I have probably more than 18 students from my lab who have gone there. It is called an Indian lab, but thanks to these wonderful students, they work so hard. I am told by my collaborator that these students are like injections to our students. They just keep on doing the work which could take a 3 week, they can finish it in 3 days. And during while this work was going on, at some stage we got a call from defense labs that dengue is a major problem with Indian Army. We said we do not work on dengue. Then we got a letter from our Honorable President Abdul Kalam that you have to work on dengue, you cannot say no to the defense lab. So, they gave us the money, and this is how we started to work on dengue. Dengue is, I think, is a newspaper knowledge in this country. So, I do not have to spend time, but we know that you get retroorbital pain, you get some fever, you can get headache, and maybe 1 or 2 other symptoms may be present, and you are okay, you are fine. First dengue is a silent dengue, you would not even notice. It is spread through a virus, through a mosquito, and for some reason, if you get it second time or third time, it becomes serious. Those antibodies which are supposed to be your friend becomes enemy in dengue. So, there is an antibody dependent enhancement of disease, and you could have dengue hemorrhagic fever or dengue shock syndrome. You start, your capillaries become leaky, you start to bleed, and you could lose a patient in 24 hours, and you have seen how much panic is there when dengue comes in India. And this is my daughter, she studies at Lady Hardinge Medical College. You can see this is outpatient department in dengue. It overwhelms the hospitals, but we blame the doctors. We say, oh, doctor came half an hour late. Global warming is spreading dengue. This is Kashmir, where there was no dengue, and you could now, the only way we fight dengue is with this, and it does not work, it just goes in your lungs. So, if you see the world map, these are the dengue areas where you have lot of rain, tropical areas, and if you can see these 3 dots. So, the diagnostic test would come from these countries. This is Australia, Korea, America, the Biorad, the Pan Bio and Standard Diagnostics. We have made the test in our country now, and we have reversed these arrows. you know. So, this is what a PhD student can do with a lens, and these tests used to be 2000 rupees, now it is 160 rupees this test. 85% of the Indian market and many other countries use this is the world leader in terms of its sensitivity, specificity. If you ever had dengue in your life, even if you do not have fever now, it will tell you that you had a past dengue or you are having dengue right now. So, it has 3 markers, not 1 marker. So, and we were told, so this is called dengue day 1 test. So, when you have dengue, you have 3 markers, a virus is there, non-structural protein, antigen and IgM and IgG, and this called dengue day 1 test, because it picks up your dengue infection from day 1 of fever, unlike many other tests where doctor will say come after 3 days or 4 days. This is, and there is a separate story which I will tell you later why is it called a dengue day 1 test. We got the order for this test from University of British Columbia. When we called them that in such cold climates, do you have dengue? They said no, we have the mosquitoes. Even if you take the mosquito and crush it with a drop of water, put it on this test, if the mosquito carries dengue, this test will tell you, and we did not know, we never tested this. These are the Canadians who tested it and told us. So, this is a great test, and the companies advertise in the newspaper saying that it is now possible to do the dengue detection from day 1. And this is my last thing to say. I was told to cover also dengue vaccine. We know dengue vaccine, if you people have heard of a doctor called Maurice Hilleman. Maurice Hilleman was a microbiologist, and we call him a father of modern vaccinology. This gentleman made 34 vaccines in his life, 34 vaccines. So, making a vaccine is not a big deal. You take a virus, weaken it, and it is a vaccine candidate. He also tried making dengue vaccine, but he failed, so he stopped. But rest of the population never stopped. We are still making dengue vaccine with live attenuated viruses, and you know the Sanofi vaccine was there. Sanofi vaccine only works in those people who already are infected with dengue. If you are naive, you never had dengue, it will never protect you. In fact, for infants, for kids, it is known to enhance their disease severity. If you take the vaccine today, after 2 years, especially in Brazil, they had seen all those kids who took this vaccine between, they were in the age of let us say 2 to 9, they had 7-fold more serious hospitalization cases because of dengue hemorrhagic fever. These were vaccine induced. So, you do not want those kind of vaccines. So, dengue vaccines have been very elusive. Why? Because it is not one virus, you have 4 viruses. So, no big deal, mix 4 viruses, but this is like an RNA virus, like hepatitis C virus, various genotypes. And okay, even that was not enough. Each virus breathes in your body, it changes its shape. This is a fully infectious virus here, and then you have non-infectious viruses. These non-infectious viruses become infectious only as immune complexes. They will not enter into the host cell unless there is an antibody attached with it. That is why when you have a secondary dengue, these non-infectious viruses become infectious because they gain entry into your host cells. So, this is called 4 serotype, multiple genotype, multiple morphologies, and they all called antibody dependent enhancement. So, immunity against dengue is a fight between, let us say, loosely speaking, police and terrorists, right. So, as long as what happens is that, so it is a balance between protective and pathogenic antibodies. Normal virus is very clever, it makes very little protective antibody, it lets your immune system make lot of pathogenic antibodies. So, this thick fat antibodies which I have shown kills the virus, but the weak antibodies make the virus go inside the cell and expand the virus. So, these are we call pathogenic antibody. So, all over the world they are taking 4 viruses, they make a tetravalent vaccine, immunize. So, you get good antibodies, these fat antibodies, but you get lot of this thin antibodies which are pathogenic antibodies. They are almost 90%, 10% are good antibodies. What we have done is that instead of using the whole viruses, 4 viruses, we use one virus like particle, a designed protein and we only make good antibodies, we do not make all these bad garbage antibodies. So, this is the difference what we have. So, we call it a dengue subunit vaccine tetravalent. It is a game changer. Why is it a game changer? You take, instead of taking the whole virus, we take the keys of the virus, the receptor binding domain of each of these virus, we link them together through our genetic engineering and express them in yeast as a virus like particle. So, this is made with dengue hepatitis, which means it will protect you from hepatitis B, dengue 1, 2, 3 and 4, pentavalent. So, you tilt it, you get huge amount of neutralizing antibodies and almost no or very low pathogenic antibody. That is the USP of our design, nobody else has that, and we have an international patent on this and in 17 different countries. We have shown, we sent this vaccine to, I am also an adjunct professor at Emory Vaccine Center. So, I sent this vaccine to them because to test them in monkeys it is not possible to do it, it takes time to get permissions. They immunized and they tell me the result that it does not work. There comes the difference, if you have done the experiment with your own things. I took the flight and next morning reached there, looked at all the results and we figured out where was the mistake. It works there too. This is where once again welcome trust, DBT, they give you this kind of ability to move and not wait on those things. And we have collaboration with Sun Pharma now, and on October 19th last year we signed an agreement to take this vaccine to phase 1, and we are working in collaboration with them to produce this vaccine in a GMP quality material. We also, I told you that we had a botanical drug against dengue, and here also we have been working with Sun Pharma, and once again we have several patents on this, and Sun Pharma is maintaining all these patents. These are our academic collaborators, from THS to AIIMS, Institute of Sciences Bangalore, NIV, CMC Vellore, University of Delhi and Rockefeller University, University of Texas, Purdue University, University of North Carolina, Emory Vaccine Center. We work with University of Helsinki, University of Turku. VTT is like an IIT of Finland. And we have had, these are our lot of industrial collaborators. Iran, Egypt use our lot of diagnostic kits, they use our hepatitis B vaccine, they use our insulin, which I did not talk about it. So, I want to thank you for your time. I showed you a lot more success, a real success, not a symbolic success on diagnostics. I truly believe that we could achieve the same success with our vaccine candidate. Vaccines are more tougher, and hopefully the botanical drug may be little easier. We know that US FDA now approves botanical drugs, and last year they approved 3 botanical drugs, all from China, none from India. So, I think we need to kind of involve Ayurveda based knowledge, and I remember Professor Valyathan was here some time back. He is a cardiac surgeon who got interested in Ayurveda. He came to ICGEB, gave a presentation on, I think his title was Ayurveda Call to Biologists. You have to provide the biochemical evidence for doing these things, and we were able to provide the biochemical evidence for doing this dengue thing. So, thank you very much for your attention, and I would once again re-emphasize the support which DBT had kept continuously. Never ever any project was ever rejected, and they not only give you the funding, they take you to those people. For example, I gave you example of Finland. So, we have funding from NIH, we have funding from Wellcome Trust, and of course, Sun Pharma. So, this is all I have to say. If any more questions, we can answer.